Well, good morning, everyone. It's really great to be here. I want to thank the organizers for pulling me up from sunny Orlando, beautiful Rochester. I also want to thank Lon Carter and Dick Wenchelbaum for making sure my question and answer space isn't going to be empty. But I have a slide I sometimes put into my presentation, and I didn't put it in this time, but I think it's appropriate since I'm representing, in a sense, a de facto regulatory agency. Some of you may be familiar with Gary Larson cartoons. There's one cartoon in particular where there's a bunch of sheep in a house party standing around holding drinks, wondering what to do. One sheep says to the other, "Henry, our party is total chaos. No one knows when to eat, where to stand, what to do." And then the other one goes, "Oh, thank God, here comes a border collie." If you had the mental image of that cartoon, the border collie is, of course, FDA. Dick Wenchelbaum reminded me about TPMT and 6-mercaptopurine, and I'll go back to 2002, and I had been following his work on the association for some time at the FDA, and I said, you know, it might be prime time to put TPMT into the label of 6-mercaptopurine. So I scheduled a oncology drug advisory committee. I asked Dr. Wenchelbaum to come up and present his evidence. I presented a proposed label for 6-MP to include TPMT, and at the end of the full-day advisory committee, the advisors voted no to include it in the label. In fact, with all these oncologists around the table, what they said basically is, look, we don't need TPMT to try to figure out how to use 6-mercaptopurine. What we do is basically look at the neutropenia and adjust the dose accordingly. Well, I thought there was a better way, so we continued pushing TPMT and 6-mercaptopurine as a prototype for updating labels of old drugs with genetic information, and in 2004 that actually happened. The other thing about 2002 I was reminded of was it was a time when genetics and genomics was not a big deal at FDA. In fact, there was very little interest in it back in 2002, even though we had Herceptin under the belt and so on and so forth. And we got a letter from pharma, and the letter, I still remember to this day, said, you know, Dr. Woodcock, who was our center director, we're really afraid to collect DNA in our clinical trials. We're really afraid to submit genomic information to the FDA. We don't know what FDA will do with it. We're afraid they might prolong our drug development programs. So could you do something about that to make FDA more genomic friendly? And we took that seriously, and over the next 2 or 3 years, going up to 2005, we created a guidance on voluntary submission of genomic data to the FDA, where we promised not to use it in a nefarious way if companies would only collect DNA and use the genomics. And we ended up getting about 100 submissions, and these were the days, 2002, 2005, that really got the ball rolling at FDA to become an enabler of personalized medicine. And I think it's done a pretty good job. During my presentation, I was at FDA actually 20 years, 16 as office director of clinical pharmacology. I was head of the genomics program at FDA. So I may say us or we, and just keep in mind that my DNA had changed after 20 years at FDA. So what I'm going to do is talk about labeling. Labeling is kind of a black box. How do we get labels that we get for products that have targeted therapies or genomic biomarkers? And these are the learning objectives. So as I said, starting in 2002, for over a decade, FDA has really been pushing the adoption of pharmacogenomic strategies. And we've done it in many different ways. It was all laid out in a blueprint where we would write some publications. We would have some advisory committees like we did with 6-mercaptopurine, subsequently with irinotecan. We developed a table of genomic biomarkers for the public to weigh in on. We had 6 different conferences co-sponsored by the DIA, Drug Information Association, to sort of get out there, what are the issues and what should regulatory agencies and industry be doing? We had a harmonization effort under the International Conference of Harmonization, and as a result of that, we have 3 or 4 guidances that FDA, the European Medical Agency, and Japanese Regulatory Agency all signed on for to harmonize the field a bit. The most recent thing on this slide is the one on the left-hand side. We put out a guidance I was involved in writing before I left FDA called Clinical Pharmacogenomics, Pre-Market Evaluation in Early Phase Clinical Studies. And the purpose of that was really to codify what we were doing over the last 10 years prior to 2013 when that guidance came out. And as you can see in that title, recommendations for labeling. So the labels are an important part of FDA deliberations. Other than approval, the next most important thing reviewers do at FDA is to look at label claims. And the label is a legal basis for prescribing and marketing drugs. It's how ads are based. It's, you can't say things about off-label use. Physicians are really able to use drugs off-label if they choose. So labels don't dictate the practice of medicine. What they do form is a legal basis for that drug. The primary healthcare provider that label is intended for is the prescriber, but obviously pharmacists and other healthcare providers read labels as well. Usually with a label, the sponsor of the new drug application or the biological license application will propose a label, and the label basically represents a summary of their early phase, late phase drug development program, biomarkers, clinical endpoints, and what have you. And then that label, usually towards the end of the approval process, is negotiated with the sponsor, and the final label that you see associated with drug packages is really a negotiated label. Label's important because it's the most common way FDA warns about safety risks and informs tertiary drug information sources like you might find in a clinic or a Mayo Clinic or a hospital, things like Lexicomp and things of that sort. All of that is drawn from the label. So label is a primary source for many of those commercial databases that are out there that pharmacists and others check. And also labels finally could be a prerequisite to third-party reimbursement. If it doesn't say so in the label, sometimes a third-party payer will say, well, we're not going to pay for it, even though that may have value in the sense of an off-label use of a diagnostic or a biomarker. Now, like drug approvals, labels are highly regulated. And this is the famous 21 CFR 201.57, revised April 2014. And you can see what I've highlighted there and underlined. The labeling shall describe the evidence and identify specific tests needed for selection and monitoring of patients who need the drug. So this was a guiding principle for labeling, for targeted therapies, for updates of label. But a second regulation that was really important, especially in regard to updating labels of older drugs, was the FDA Amendments Act that came out in 2007. And it redefined, in many ways, the standards for efficacy and safety. And I've taken one sentence from that Amendments Act, and it says, safety is defined in a way that allows efficacy failures to be treated as a safety issue. Now, when you remember, or you may remember FDA updated the label of Plavix or clopidogrel. And the reason for that was 30% of patients are so-called resistant to the drug because they can't convert it to an active metabolite because of their 2C19 polymorphism. Well, that was a safety issue. It was a lack of efficacy issue, but it was also a safety issue. And the same principle was applied to the labeling of cetuximab and panitumumab for colorectal cancer with the KRAS, because there are certain people with KRAS mutations that could not respond to the drug, and the lack of efficacy became a safety issue. And this was one of the pillars, along with the previous regulation, that underpins labeling with genomic biomarkers. Now in the FDA, there's a lot of negotiation about labels, and part of it is driven by the data, the science, as I say, but a lot of it is by judgment and how people look at the science. So the questions that come up in labeling a drug product are, what are we going to include and not include? So if you have a 2D6, do you put the genotype in there in the label or the phenotype? Do you put the specific mutant alleles in there or what? That's a question. I had irinotecan as an example because in that case there was a UGT polymorphism that caused a significant toxicity in terms of neutropenia. And there are other similar alleles that were never tested in humans, but there was plenty of animal data that says they are the same type of allele, will have the same effect on toxicity of irinotecan. But we were not allowed in 2004 to put those in the label, even though they were not, they were at-risk genes, because there was no human data. That's no longer the case. Where to include it? Clin Pharm section, DNA. Certain sections of the label we know are read more than others. Indication, dosing and administration, contraindications. And I'll show you a label where they're all pulled up to the top of the label in recognition of their importance. What type of language? genotype, phenotype. And then where do you cross-label it with other sections? So every label, there is no like national standard for labels, although FDA is moving in that direction, like a can of spaghetti where you read a barcode, or you see the nutritional information on a food product. There is no national standard for labeling, so every labeling decision becomes unique. That's changing in the sense of the physician labeling rule, and this attempts to set a national standard for content and format, what to put in a label, where to put it, and how to put it. And I've shown a chronology of the physician's labeling rule, and you can see that there are various guidances that are worth checking out to find out more about pharmacogenomic biomarkers and what's allowed to be put according to these guidances in the adverse reaction section, the DNA section, warning and precaution, and so on and so forth. So if there's a question about how does stuff in a label get into contraindications, these guidances will help you understand what the regulatory thinking is on where to put the information. Now, this is an amazing label, and I show it at this point for a number of reasons. First of all, the genomic biomarker is cytochrome 2D6. It's a metabolism polymorphism. It's a pharmacokinetic, it's a metabolism pharmacokinetic combination. And what's amazing is that this drug called luglastat was approved in August of this year for the treatment of Gaucher's disease. Well, if you look at that label under indications and usage, you can see that there's something about 2D6. And what it says basically is that before you give this drug, a priori, you should test for 2D6, because if you're an ultra-rapid metabolizer, you shouldn't get this drug, or at least it shouldn't be prescribed. Furthermore, if you're a poor metabolizer, the dose that the patient gets is 50% of what the extensive metabolizer gets. 2D6 appears in limitations of use, the DNA section, and the contraindication. And it's amazing in the sense of what I said about 2002 and TPMT and the difficulty and challenges to get these kind of standard pharmacokinetic metabolism genes in the labels that we have today. In 2014, a drug that was approved, which mandates, I shouldn't say mandates, which highly recommends, if not requires, a 2D6 test be done for this particular drug. The test, typical of FDA policy today, needs to be an FDA-approved 2D6 test. And the reason that this got so strongly worded in the label is that the manufacturer who developed the drug tested all of the patients in the pivotal clinical trials using 2D6 before they got the drug. So the clinical utility, the clinical validity, et cetera, were all confirmed during the drug development process. The other concern with this drug was QTC prolongation. The risk of QTC, because it was related to serum levels, was closely linked to the genotype of the patient. So there's two categories of genomic biomarkers that impact labels, and they're both in a broad bucket. The first is the test developed in association with the drug. That's represented by that previous example. This is the least challenging in terms of labeling of a drug product. And the reason is that rigorous randomized controlled trials are done during the drug development process, so they get all components of a drug test combination, utility and analytical validation. The endpoints are clinical approvable endpoints, not surrogates. And because of that, it's easier to label. Both enter the practice together, so the drug and diagnostic go together. There's not a lot of re-engineering of the medical practice when you have a situation like this. And finally, it's a less burden on adoption because the drug and the test come out and all the educational materials, advertisements link the two together. Tests offered after the drug reaches the market is a whole different criteria. This is the warfarin, the TPMT, the 6-MP, the clopidogrel, and all of those drugs that Dr. Weichbaum had on his slide. Greatest challenge for labeling, and the reason is that the evidence base is not as good as the first case where you have the drug and test developed together. In labeling these products and updating them, they were generally based on observational trials, in some cases small n's, but it was collective evidence. In the case of warfarin, we had over 15,000 patients indicating that genotype matters in selecting the dose. The soft endpoints were a problem in updating. With warfarin, for example, most of the data was based on INR. Yes, physicians use it in clinical practice, but that's not the clinical endpoint if you were developing warfarin or an oral anticoagulant today, bleeding risk would be the endpoint. So there was a soft endpoint, and the benefits were not as clear. Because the drug was in the market already, physicians were comfortable with it. There usually were alternative approaches to personalizing the therapy, and therefore adoption required some change in the practice. The little asterisk on that slide is a recommendation of using Hill's criteria for causation, which is what we used at FDA and is still used for FDA. These are 10 criteria that link A with B. So when you're talking about Hill's criteria, you're asking the question, is there a dose response in terms of relationship between drug and effect with that genotype? Is there a mechanistic basis? Is it reproducible? And while this has been generally applied to pharmacoepi studies, we used it at FDA to determine whether the Hill's criteria was met, and therefore that furthered the adoption into a drug label. Now, I put this drug up there because Herceptin was an efficacy biomarker, approved in 1998. We wrung our hands at the time when this was under review because there was not a good diagnostic at the time of approval. The to-be-marketed diagnostic came a little bit later, and there was also some question about whether patients who expressed +2 received any benefit. In fact, they received very little benefit in the pivotal trials that was part of the submission. I put it on here, though, because it was the first co-developed drug test combination that FDA approved in 1998. It was also a good example of what FDA calls today breakthrough therapies, because the review time for that drug was in line with breakthrough therapies that we see today. So in many ways, 1998, it was a model for drug and companion diagnostics that are being developed today, and many of them follow the same scheme that Herceptin used. This is Mellaril. It was the first approved drug for which we went back and relabeled it. And we relabeled it with 2D6 because there was a high incidence of prolonged QTc and 2D6 poor metabolizers, and we put this into the contraindication section of the label and followed the same pathway of all of the drugs we've talked about. So co-development is the ideal situation for planning for a good label. 12% of all drugs approved in the last 14 years had information on the label at the time of approval. It's not as many. I expect that's going to go up with time. 18 cases through October 2014 where the label directs clinicians very specifically to use a diagnostic test. No surprise, many of those are in oncology. There's been a rapid rise in PGX information and labels, and you can see every 10-year period there was quite a dramatic rise. In the beginning, in the 1986 to 2005 period, most of the biomarkers in labels were pharmacokinetic genes like the 2D6 and so on. And then in the last couple years, we've seen many more biomarkers that are being proposed in drug development because of targets, as we've heard today, and because of pathways. The types of biomarkers has changed over this time period. Again, PK and CYP gene variants were very common in that 10-year time period. We tried to use those to identify a dose. Later on, as Dr. Carden mentioned, immunological gene variants came into play with regard to safety and immunological reactions, and these were biomarkers that were put in labels by and large to avoid taking a drug and to avoid toxicity. And then more recently, in the last 10 years or so, we've seen that biomarkers in labels have really focused on targets and pathways that are used to select and identify a drug for subsets of the general population. So there's this changing climate, and I suspect we're going to continue to see this evolution with more and more efficacy biomarkers leading the way, with some of the other biomarkers coming along for the ride. Now, when it comes to drug labels, this is something that Dr. Carden alluded to in his Q&A. Most of the actions you can see is in oncology with the target pathway biomarkers and labels, and in psychiatry. Almost all of the drugs, there's probably 18 or 20 drugs in psychiatry that have labeling with the 2D6 biomarker. And there's enough clinical outcome data now in psychiatry that's begun to show that a priori testing with 2D6 enables the right drug to be selected for the right patient, and then clinical outcomes follow that with improvements in clinical outcomes. So right now we have 141 drugs with 45 genomic biomarkers in the label. Almost half of them are these metabolized or transporter genes, the 2C19s. 30% are the targeter pathway like the HER2, the BRAF, and 23% are immunological, predominantly the HLAs. So regulatory policy doesn't happen overnight. It sort of evolves. There's regulations, there's precedent decisions, there's guidances, and then that's where FDA comes down. And what I've shown here are some pivotal decisions that occurred over the last 10 or 15 years that I was involved with that shifted regulatory policy with regard to PGX biomarkers in the label. HER2, again, first co-development case, I mentioned it. The challenge in the review of that was the LDT that was developed for Herceptin, where the FDA wondered whether they needed to approve it with an approved diagnostic or with an LDT. Thioridazine, I mentioned, it was the first time we put a black box warning in a label with a genomic biomarker, predominantly for a safety reason. 6MP was the first label that used a genomic biomarker to stratify the dose after you get the TPMT gene allele analyzed, going from a low to a high dose, depending on whether you were the 0.3% poor metabolizer or the 11% intermediate metabolizer. Panitumumab was interesting. It involves the KRAS gene. And this was the first time FDA put in the label to test for KRAS to really prevent people from getting it that had a mutation. It was a prospective, retrospective study in the intent to treat population. It was originally approved for all comers, and after some reanalyses and some advisory committees, it was found that there was a negative predictive marker in KRAS, and the product was relabeled. So this was the first time that retrospective looks at that data was allowed. And there were some criteria that allowed that to happen, such as a high ascertainment rate, which meant a high percentage of DNA was collected in the clinical trials, a good analytical method for KRAS, an a priori analysis plan, and things of that sort. It wasn't just willy-nilly, but very definite criteria. Abacavir was a poster child for drug safety. It was the first post-marketing commitment randomized controlled trial that led to a black box warning for people that were positive for the HLA B-star 5701. And it was one of two questions that the FDA had about this product. The other had to do with cytochrome 1A2 as another contributor to the interaction, but to the company's credit, a randomized controlled trial was done. I don't think that's ever been done since for a safety question. Tetrabenazine is on there because it's labeled genetically with regard to poor metabolizers, 2D6. But the interesting thing about tetrabenazine, and it's now carried over to a recent approval for Brilinta, is that no genetics was done in the study as the drug was being developed. Rather, drug interactions were used to label it with genetic information. So for example, if the company did a 2D6 inhibition drug-drug interaction study, and in a sense, created a poor metabolizer, that filled in for and substituted for a formal genetic study. And there's now been a handful of drugs that have been labeled that way with genetic biomarkers in the face of no genetics being done during the clinical trial, but the data is based upon the drug interactions that were occurred. Warfarin was a very famous example. It was the first detailed genotype guided dosing. If you look in the label of warfarin today, there's a very nice Excel spreadsheet. It was labeled 2 times, 2007, 2010, and in some cases it beat out standard of care in getting people in their INR quicker. Vemurafenib was one of the fastest approvals in FDA history. It was the first to describe a specific FDA-approved test in the label itself, and that trend has continued. The review time for Vemurafenib was 4 months, which was a real prototype for the breakthrough designation that FDA is now using for very effective therapies compared to standard of care. And the last one on there was ivacaftor. That was interesting because it was the drug that first got breakthrough therapy designation by the FDA for the 5% of cystic fibrosis patients that are G551D determined. And that was approved without a companion diagnostic because in the transplant community and in the CF community, the 510K assay was already available. So it was one of the most rapid approvals in FDA history. What this slide shows is the informational value of PGX biomarkers in the label. It combines the FDA and EMA databases. 191 drugs approved by both agencies have pharmacogenetic genomic biomarkers in the label. 94 of those are in FDA only, 25 in EMA only, and together EMA and FDA has 72. What's important on this slide is the informational value, and you can see from these pie charts that genomic biomarkers in the label 35% of the time were there for information value, usually in the clinical pharmacology section of the label, and most people more or less ignore that. 40% are actionable pieces of information in the label, but there is no specific test included in the label. Somebody would have to look at that information and interpret it, a clinical pharmacist, a genetic counselor, and say, oh, that's actionable information. Let me interpret it and translate it into genetic testing. 25% of labels do have actionable information, meaning that the test is either required or recommended. Now, much of the information on the metabolism transporter genes, I mentioned almost 50% of labels have this information in them, ends up in the different sections of the label. We have 70 drugs and 10 CYPs or transporter gene variants. Almost all of these drugs are either 2D6 or 2C19 as their genetic alleles, and 30% of those are actionable. About 2/3 of the labels that have these biomarkers recommend a dose reduction, similar to, say, clopidogrel or warfarin, and 1/3 recommend or avoid use, as does the 6MP label. Now, this is an interesting part of FDA labeling of products. It's often a question that people ask about, why don't you just require a test on a label? And I'll just go through this pretty quickly in a rank order sort of way. When KRAS and cetuximab were relabeled with KRAS predicting a negative effect, there was a strong gene outcome association, there was replication. The label basically says test everyone before prescribing the drug to be positive patients, exclude the negatives. And there was a subset effect. TPMT and 6MP uses a different language, recommended. Reasonable gene association outcome, the label says consider testing. And the reason it was consider is because at the time it was relabeled, there was some uncertainty about the exact dose that should be given to poor and intermediate metabolizers based on the TPMT. We knew it was a good idea to lower the dose, but it wasn't specific enough to say give this dose or that dose. VCOR and warfarin, these are an example of actionable biomarkers. As I said, there's a table in the label that is 2x2, 2C9 on top, VCOR on the bottom. You match up your patient, and you go from 5 milligrams, or excuse me, 15 or something milligrams at the high end, depending on the genotype combination, down to about 1.5 milligrams on the other end, depending on their sensitivity with the VCOR genotype and their rate of metabolism with the 2C9. The label, though, is silent on requiring or recommending the test, but an informed user can obviously use the test. 2D6 and fluoxetine, we relabeled this over the years, and this was really informational. It's an example of an informational label. 2D6 poor metabolizers had 30% higher adverse events. Most of them were not serious, and therefore testing wasn't recommended or required, but one was instructed in a label to monitor therapy with the drug. So why aren't all actionable genomic tests and labels required or recommended? Well, first of all, the test is implicit in indications in the contraindications section of the label. It might say, for example, that clinical trials that were pivotal included this gene as a selector gene for a responsive subset. That was the case in that drug I showed you early on with the Gaucher's disease. Even though it's indicated for certain genotypes of 2D6, it doesn't say in there test for 2D6. And the reason is it's self-obvious. Sometimes there's an absence of prospective randomized controlled trials as an evidence base. We would have said a stronger statement in the warfarin label had there been a randomized controlled trial at the time, but the evidence base from the medical side was considered to be relatively weak because there was no randomized trial. Third, lack of outcomes in biomarker negative patients. When FDA approved Tarceva years ago, it had a Kaplan-Meier plot that showed a very nice response in biomarker positive patients, and biomarker negatives were almost like placebos. When the drug got to the marketplace, further evidence said that biomarker negative patients might respond to this drug, and the label was changed back to the fact that it could be used in all comers. I think it has since been changed back to EGFR positive patients. But this lack of outcome in biomarker negative patients is often why you don't see require the test, because there's a hope or an unknown, an uncertainty that the drug may work in negatives. Inability to interpret test results in some genotype is another reason. Lack of access to the test by healthcare providers. The feeling was it wouldn't be fair to say this test is mandated, but not have it widely available for physicians to utilize. And finally, there was some concern from legal side that putting a required test in a label could set physicians up for lawsuits if they fail to use that test in a patient, even though it might have been warranted for medical reasons. And indeed, now we're seeing lawsuits come about in 5 different states on Plavix because patients have been harmed by not having their 2C19 measured. I mentioned this Brintellix that was recently approved for depression. And this gets us into an area that's relevant to labeling, and that's phenoconversion. Phenoconversion means that one could create a poor metabolizer phenotype, either from a genetic standpoint, if you have a certain allele, or from a drug interaction. The point of this is that phenoconversion to poor metabolizers occurs at a rate of about 25%, for example, in the depressed population. Whereas genotype, poor metabolizers, generally in Caucasians is around 6 to 7%. It's important to understand this for a couple of reasons. One is that genetic testing for 2D6 alone may not identify in clinical practice all of the 2D6 poor metabolizers. So one has to think about polypharmacy, what the patient's taking. Are they having a drug interaction that converts them to poor metabolizer status and not be misled by genetic testing? The second thing is, as I mentioned, labels have been updated with genomic biomarkers based on drug interaction studies. Vortioxetine is the most recent. It's up there in the highlights section of the label. And recently, Pimozide, which is a drug that's not widely used. It's used for tics in children in particular. This was labeled with genomic biomarkers recommending 2D6 testing because single-dose PK studies showed a difference in the exposure to the drug with the different phenotypes, extensive, intermediate, and poor. And the label, if you go to read it, recommends that testing is done based on this pharmacokinetic study, and the actual results of those studies were simulated. In other words, people took pharmacokinetic studies at FDA and simulated what would it look like if I gave this dose, the usual approved dose, to poor metabolizers versus extensive metabolizers. How do I compensate for that difference in exposure by recommending a lower dose? Again, it's an example of a surrogate, if you will, for genomic biomarkers in the label. Now, FDA has created, I think, a very nice framework for the regulation of genomic biomarkers. And as you can see, starting in 2005 with the guidance on how to submit pharmacogenomic information, it's accelerated very rapidly over the last couple of years. Just this year, probably 2 or 3 guidances are on the street looking for comments from stakeholders. FDA is holding public meetings and what have you. What's coming up is a guidance on drug diagnostic development. FDA had one of these guidances back in 2005 and 2006 that the industry said basically was unrealistic. It never really got out as a formal guidance, but it's been revisited, and there'll be a draft on the street within the next couple of months. So this is the sort of compendium. It's like the Goodman and Gilman for pharmacology, only this is the FDA's book on regulation of genomic biomarkers and their impact on labeling of drug products. So in summary, the take-home messages for me is that the regulatory framework, unlike that party I mentioned in the beginning where the collie showed up at the door, I think FDA has done their job, not without controversy. I know they've worked hard to create a regulatory framework for industry, and I think it's begun to mature. It's multifaceted. There's great cooperation between the Center for Devices and the Center for Drugs. And I think it's very transparent in terms of 2 things. One is the guidances, and secondly is the approvals that are made, especially with companion diagnostics or co-developed drugs, because the summary basis of approval is online at Drugs at FDA, and anyone could go read what the reviewers think about that development program. The co-development, I think, is the future for the most part in individualized medicine. And what it really has done is refocus stakeholders, the company, clinicians, FDA, on the importance of clinical utility. Is the biomarker fit for purpose? Does it make an incrementally large enough difference to include in a label? And what's the evidence to show that? And finally, the approval of companion diagnostics in the market will be challenging. Putting a companion diagnostic out there for an approved drug is going to be a lot more difficult than it was in the era I was at FDA because of the higher evidence standards, and the exception to that would be a very serious adverse event where a label is most likely to be updated, irregardless of what's out there in companion diagnostics. So with that, I'll close, and I think I got 20 minutes to spare.
Gosh. Well, that just means more questions. Come on over. I think it was only 20 seconds to spare, but.
That's what I meant. I wanted to see if you were listening.
I was listening to every word. Now we're going to see if all of our audience was. Thank you, Larry. I just want to pick up on your closing point as I was listening to it there with respect to companion diagnostics and some of the challenges that you see us ahead. Because I do think, and I'll speak from a layman's perspective now, there's been a lot of interest in that area and optimism, but you were kind of getting at some of the challenges. Could you just elaborate a little bit more on that?
Well, I think the optimism, I mean, if I'm on the outside watching FDA as a consumer, I think the optimism is around the number of drugs that are coming out with drug and companion diagnostic. I think we're seeing more in 2013, 2014 than we've ever seen. And I think we're beginning to see beyond cancer. And most of the action has been with oncology patients, but now we're beginning to see in infectious diseases with hepatitis C.
Right.
We're beginning to see a lot more development in the area of psychiatry. I think there's a lot of companies with drugs in psychiatry in their pipeline that will designate subsets of the population with serious depression or schizophrenia that will help bring some higher efficacy rates than we normally see in those diseases that normally run 30 or 35%. So I think we're going to see an expansion of the therapeutic areas in which we'll see drugs and companion diagnostics, which obviously will affect a whole bunch more of the American public. That being said, there's some challenges in other chronic diseases. For example, companies have tried and not really been successful in getting companion diagnostics and drugs for obesity. Diabetes is another area. So I think we have some really frontiers to think about, and I think we have to look at the genetic underpinning of these diseases and, in fact, what percentage they would contribute to the disease itself rather than environmental conditions of diet, exercise, and all that sort of stuff.
Okay.
So I think that's kind of where we are. Great news in oncology. Coming up fast is infectious disease and psychiatry, and then later on are some of the more chronic diseases where this would be very beneficial in terms of improving efficacy rates.
Okay. Great. Several questions, as we expected, so thank you, folks. Let me start with one, again, perspective of FDA and how they view pharmacogenetics or genomics, I suppose, PGX, biomarkers as being validated or exploratory.
Well, we had to, when I was at FDA, make that distinction because companies would often say, do I have a validated biomarker, and therefore can I use it for decision making in either clinical trials or in my label to recommend for physicians? So we created a very simple hierarchy. The validated biomarker had the following criteria. One is you have to have a very robust analytical system, analytical validation, clinical validation, ready to go to the marketplace. And number two, the evidence base was very strong. The biomarker was validated with clinical evidence, not within a single company necessarily, but well accepted in the outside world. So for example, when you take things to an FDA advisory committee and you get a vote of 12-0 or something like this, that this biomarker is acceptable, that's going outside the realm of a single company. Furthermore, you have drugs with multiple indications based on HER2, for example, or multiple drugs based on a single target from different companies. There you have a public sort of validation of that. That's validated. The other area is what we call probable validated biomarkers. And I know it sounds like a little wiggle room there.
Sounds like Washington to me.
But probable validated was something we called biomarkers that were developed within a company and in all likelihood looked like they were validated, but it needed further expansion into the public domain through an advisory committee or something like that to get a blessing of and a confirmation of that validity.
So sort of steps along the way.
There were steps along the way, and then everything else fell into the third category, which was exploratory biomarkers, and they were generally interesting. They might have been helpful internally to a pharmaceutical company to make a decision on strategy, but they weren't ready to go into a package insert.
Some really good questions here, and I'm going to read one that came in from the audience. What are the regulatory implications of the new FDA LDT as Class 2 and 3 medical devices and methods? And this person says, does the healthcare system need to follow the quality system?
Yeah, boy, you're getting into some swampy areas here. Mainly because these are areas under active discussion at FDA, with Congress weighing in as they normally do, and all of the professional societies weighing in on what they think about LDTs and so on. It was only about a week or two ago that the FDA released a draft guidance on LDTs. So anybody that wants to read the nitty-gritty of the perspective on LDTs, go to the FDA website, September 30th. The guidance came out.
Can you give us the Cliff Notes?
Yeah, the Cliff Notes are as follows. 2 ways to get a biomarker to clinical practice. The first is the in vitro diagnostic way. It's the regulatory pathway. There's a pre-market review. And the quality of that test is by regulation covered by Quality System Regulation 200.1. So it implies a higher quality because FDA sees it, they review it. It generally ends up in a label as a trademarked name. And that's the in vitro diagnostic path. Higher clinical evidence, perhaps, higher quality analytical. FDA sees it before it gets into the label. Okay, that's the in vitro diagnostic. That would be, for example, the HER2 example with Herceptin, or some of the more recent examples. The other side of that ledger is the lab-developed test, sometimes called home brews. Home brews, lab-developed tests, are usually developed in a single laboratory. FDA doesn't see that test. It's covered by CLIA quality regulations called Quality Management System, QMS, and it generally has to reside in that laboratory in which it was developed, and it can't be generally distributed throughout the country. So LabCorp or these other couldn't distribute it everywhere. Now, what FDA has proposed is to regulate LDTs, to the angst of a lot of diagnostic companies. And there's advantages and disadvantages to that have been laid out, I think, in the press quite nicely. But the approach that FDA is taking is realizing they can't possibly oversee all of the lab-developed tests. There are just hundreds, if not thousands of them. And therefore, what they've taken is a risk-based approach, as they call it. There's 3 risk categories, and the high-risk category would be virtually anything we've talked about at this conference. The CYP enzymes, the HER2s, the BRAFs, and so on. Anything that goes along with a drug from which a physician makes a decision to give the drug, dose the drug a certain way, or prevent a patient from getting the drug will be a high risk, and they will be the LDTs that will be covered first. The rest of them are going to be phased in, probably being realistic over 10 years. So that's kind of where we stand.
Okay. A couple of questions following on your discussion about the labeling. So there's one here that talks about, and you suggested the potential for some new national standards for labels. But this questioner asks, couldn't that add additional cost to both pharma as well as the regulatory agencies? And could that in turn elevate the price of drugs?
Yeah, so the physician's labeling rule was intended to make labels identical from one drug to the other to the other. The label is a repository for the information that was generated during the drug development process. So in that sense, it's not changing the requirements to put information into the label. What it really is doing is giving some rhyme or reason to where information will go. For example, if you look at today's label, and I showed 1 or 2 of them, you have the highlight section. If you didn't read the fine print, which is the rest of the label, and I've been told people don't read those labels. If you read the highlights section, as you said, it's the Cliff Notes for using that drug properly, and I think that has been a big boon to improving drug labels. Patients even like that highlights section because you can get boom, 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 and you get your information very quickly. The internal decision that companies and FDA negotiate is what goes in the highlights or not goes in the highlights, particularly if it's bad information about a drug. Does a precaution or a warning about an adverse event, is it serious enough and of the highest prevalence to move up into that highlight section where it gets more prominence and more attention? And that may not always be desirable. But from a cost standpoint, you know, unless you're feeling pressure to make sure that all of the boxes and the highlights, drug interactions, whatever, are fully informed, and therefore I better do more studies, the labeling rule in and of itself isn't necessarily going to increase the cost of drug development.
Good point. Another one here, a comment and then a little bit of a question again on the drug labels. So this audience member observes that drug labels are a rich source of information, but they're only available as human readable text. The structured product label make the content available electronically, but there is variability in how the sections are populated. So the questioner asks if the FDA is working on a more structured format that you could essentially use this data for informatics research.
Yeah, that's a good question. Labels come in electronically into FDA, as do the NDA itself, New Drug Application. And labels are online electronically in some of the third-party companies that take these and put them online.
Right.
I can't name them all because that's not an area I'm familiar with. But I think it's important that the source of that label has something that's searchable.
Right.
And something that is consistent in the way they lay it out. So there's a lot of third-party databases that are based on FDA labeling that are electronic in nature. And there's probably, you can check that out with a Google search, but I don't know the brand names. We use Lexicomp in Florida, for example, that has a lot of electronic information in it. And it's highly, let's say, consistent with the FDA labeling. That would be another thing I would look at. There's other ones out there like Hippocrates and so on. And the thing to look for in these databases is how inclusive are they of the information that's in the FDA label? And how is that information presented accurately? And that's worth self-assessing if you're getting into that kind of electronic format.
So this question appears a little technical, and we might want to just explain it a little bit to some of the folks in the audience. But the questioner asks, if I get new approval as a Class 2 medical device with human genome reference, HG19, would I need to get a new 510K every time the reference genome changes? So maybe give folks a little context for that question if you can.
Well, I'll try, but that comes from our Center for Device perspective.
Okay.
I'm a drug guy, Center for Drugs. I don't know if I know the answer to the question, but it reminds me of FDA-approved tests that make their way into the label. We've had this situation before. We had it with the Herceptin in the early going, where there was a IHC test that later went to a FISH test, and that was an approved test. And then the question was always, what happens if I want to make a change in the approved test to make it more efficient, faster, cheaper? Do I have to go through the whole rigmarole of getting another approval through a 510K or something like that? And I think the answer to that question, really, it's case by case, and it resides in the change that's being made from what was previously approved, how significant that is, and what degree of cross-validation was available for that.
Right. Final question, maybe a little bit out of left field, but really picking up on a number of our conversations at this conference yesterday and today around cost and the cost of medications. And we had a presenter yesterday talked about cancer drugs in particular, how sky high they are. Do you envision a time, I don't know, within the next decade or within our lifetimes, when the FDA will start to consider cost in its deliberations? And do you think that that should be occurring?
Well, that's a good question. I would say, in my experience at FDA, cost was not something FDA considered, and well, they should. They have a regulatory mandate, it's written in law, it's interpreted through guidance, and cost does not come into that consideration. You could argue, though, new regulations that could possibly increase the cost of developing, say, a companion diagnostic should come into consideration, perhaps. Not so much in the scientific content, but in the judgment of what would be covered by a regulated device, for example. So you could say, well, okay, taking LDTs in the high-risk category and making them in vitro diagnostics could increase cost. And I think that's something that needs to be considered. But in terms of the approval process of once that decision is made, costs would not come into play.
And do you think that that should really remain the case, that FDA sort of stays within its parameters?
People are human. And I always like to say at FDA we dealt with science, which are the facts around the medicine, and then there was the judgment call.
Right.
And sometimes the judgment call comes into play in, for example, the incremental benefit of a medication. Not that it would not be approved necessarily, but how it might be labeled would be an issue, and the claims that would be allowed in the label. Even some of the unknowns from the drug development process could appear in a label. I could see that in an indirect way affecting cost, if you will.
Sure, so if you have the second or third of a therapy,
Right.
it might make that observation about this being an incremental improvement.
Right, or if I decide to take something to an advisory committee as an FDA official or not, could have a bearing on what happens with that drug and development program as well. If the benefit is huge, I don't think there's an advisory committee on the horizon. If it's marginally better, I think some questions would come into play. Is the benefit risk, which FDA uses for approval, is the benefit risk adequate? If I have all of the downside of the drug but not much incremental benefit, should that drug be approved? And that indirectly, I think, affects the cost of medication.
Great. Well, I know folks were eager to get all of these questions to you, Larry, and so let's thank Larry for answering them all.
Thank you.
Thanks.
Appreciate it.